Welcome to the lab instructions for our phylogeny lab. But first, we're going to start with the answers to the basic phylogeny lab that you all did last week. So you had to make a phylogeny, including all of the groups that you see here on the screen. And many of you made good phylogenies, some of them pretty correct. But we're going to look at some details as to what you might want to consider as you make a phylogeny out of these groups. So there's lots of different ways you can represent the phylogenies, and I'm going to show you a number of them, including two that I made myself, and then a number that I took from different websites. But there are a few non-negotiables for this. The first is that the jawless fish need to be their own branch. So everything else is going to be kind of separated from the jawless fish. We'll have them coming off first as their own branch. And then we'll have the sharks and the chimeras also on their own branch. And then from that, we'll have a branch that includes the ray-finned fish and the lobed-finned fish. And then coming from the lobe-finned fish branch, there will be another branch that goes to the amphibians, the caecilians, and then salamanders, with frogs coming off of salamanders, or later than salamanders. There will be the, this is really interesting, the synapsid branch with the mammals. I'm not really sure what happened there. And then a branch with the turtles and the sauropsids. Sorry, that sauropsids is apparently the meal of mammals up there. And then lizards and snakes will be off of the sauropsid branch, and then crocodilians, dinosaurs, and birds on another sauropsid branch. So let's look at one of the ways that I represented this. So this is one of the possible ways you could have represented this phylogeny. So you can see the jawless fish, the sharks and the chimera, and then the ray-finned and the lobe-finned, eventually giving us all of these other groups as you see here. Synapsids and sauropsids, I used arrows to indicate where those would have been. But here is another way you could have looked at it, and you can see the different groups and some other groups that I didn't include in here. Another different way of looking at it, this one including some pictures. My favorite part of this perhaps is the reptiles with the picture of a duck, and then including birds. Here's another way, and this one was actually looking at, as you can see, the aquatic versus terrestrial portions and the number of species in each of these groups. Another way of looking at it. And then finally, once again, this is one of the creations that I had done with it. And none of these perhaps is really perfect, but they all sort of give a very similar answer. And with this one, what would have been before the jawless fish branch would have been the first vertebrate. So we're looking at how do all of these fit together well, which is something that you will be doing in today's lab. So for today's lab, the phylogeny lab, we're actually not doing it today. We're going to be doing it on Wednesday and Friday in place of lecture. The lab report is still going to be due November 27th at 2 p.m. via Brightspace. You could start working on the beginning portions of the introduction even before we do this lab. So before I give the instructions for this lab, we're going to go over some basics to hopefully remind you of information we learned all the way back at chapter 1. So what is a species? A species is a population that is reproductively isolated from similar populations. If you remember, it's hard to figure out exactly what a species is as we look at things like fossils, because fossils cannot breed. We usually use reproductive isolation, however, to identify a species, but reproductive isolation doesn't equal genetic isolation. And we find that as we move animals and as we domesticate animals, we can actually alter some ideas of speciation. We use type specimens or holotypes as representative descriptions for species. And then we talked about the idea of what if they're not accurate. And that was when I mentioned that the creature on this slide is a fox squirrel. He is our regular tree squirrel around here. And this is what most of us would say one looks like. Some of you might say that they're more gray than they are red, but the holotype that is kept for this was actually a black representative, which is really problematic because black fox squirrels are rare. I have seen albino fox squirrels, also remarkably rare. Those are not good type specimens for this. However, we've discovered this in some other species too, that our type specimens have not been good representations of a species. So then how do we tell a species apart? Well, we look at characters. These are your traits that you use to distinguish taxa. Ancestral ones were present in the ancestors. Derived characteristics are ancestral characteristics that have become modified. For your vertebrates, your ancestral characteristic generally is the vertebral column. Things that are very derived in different groups include foot and hand bones. We use these derived characteristics shared among different groups to try and figure out relatedness and then trace back towards a family tree, looking at the combinations of derived and ancestral characteristics. By doing so, you are taking part in phylogenetic systematic work. You're classifying things based on their similarities. This can also be dealing with cladistics, as you classify things according to their clade or their evolutionary lineage. And we're going to look at how things are related by looking at their structures, and you are going to relate them by common descent, by shared derived characteristics. You're also going to look for some plesiomorphies, or the inherited unchanged ancestral characters. You will remember, hopefully, that word parsimonious. You're going to try to create the most parsimonious phylogeny you can by having it have the fewest number of changes to get to each step. Because you should remember, I hope, that a change in structure is an unlikely event. And you should always, when you're looking at things from an evolutionary perspective, aim for a phylogeny that has the fewest number of changes necessary in it. These phylogenies are hypotheses for evolution. We can test them, and we do test them, and you will be testing yours using primary literature. But how do you know it's ancestral? Well, by now you might know, because we'll be using representatives of living creatures to try and figure this out. But you might want to look at the largest number of species and figure out how things fit together while looking at in and out groups. We went through this activity in class, and when we went through the parent at the top and then, or the most ancestral at the top and then the more derived there below, we looked at these, and you may want to pause it here to go through this again to remind yourself, but we decided that A is the more parsimonious and that B and C were probably not correct. A requires only 3 changes, whereas B and C each require 4. We then used this example, and we built a phylogeny after determining that the one with legs and stripes was the ancestor. We built this possible phylogeny. So you're going to remember the idea of parsimony, and you're going to think about evolution. What is likely to occur? What is easiest to recur? And how are these different groups related? We're now going to follow an example that was listed in the lab manual. In this, I received a packet of cards with pictures of a shark, a skate, a tuna, a sardine, seahorse, frog, salamander, tuatara, T-Rex, eagle, sparrow, platypus, kangaroo, mouse, elephant, and a whale. In case you're wondering, is there one with this exact group? No, thankfully not. But you might be wondering, okay, this is a mix of different creatures. Some will be like this. Some will be a fish group. Some will be a mammal group or a bird group. There's a couple that are reptiles with amphibians. And then there's a number of these mixed groups. You're going to have to do one mixed group and one type of single group and likely one other one of your choice to create your different phylogenies. What I would suggest doing when you receive a mixed group like this is to first divide them up into their groups. Divide up the fish, amphibians, lizards, dinosaurs, birds, and mammals. And then you may want to subdivide inside of this, the cartilaginous versus the bony fish, frogs versus salamanders, the different types of lizards, and see how things might go together. You want to then figure out what is likely the oldest of the groups, and then the relationship between the rest within the groups. Then you put the groups together to figure out their relationships, and you build a family tree. So here's one possible way of looking at this. And for the most part, it makes sense. We have the shark and the skate together, they're cartilaginous. The sardine, the tuna, and the seahorse, while in this, I assumed that the sardine was likely earlier because it's a small fish, and that the seahorse was later because of all of its more derived features, and the tuna in between. I then put the salamander and frog on their branch, the tuatara on its own, because I had no other lizards, the T-Rex on the same branching as the sparrow and the eagle, the platypus and the kangaroo, then separate because one is a monotreme, the other is a marsupial, and then the mouse, elephant, and whale as the rest of the mammals. So if you're looking at this, you're probably realizing there's likely to be one big mistake here. The frog and the salamander should have been switched, because we would assume that frogs probably came later in the record, even though for the individuals we're looking at, we may not be able to know with what we do know. So what are you going to be doing? You're going to be working in groups of 2 to 3. You're going to be creating, right now, we're going to say 3 phylogenies, one specific group, one mixed group, and one of your choice. Once again, note that reptiles, turtles, and amphibians are together in one grouping of pictures. You're then going to either draw or write out the phylogenies you've made, and you're going to write down some reasoning as to why you made the decisions that you did. You're going to hand in your drawings and your reasonings at the end of class on Friday. You will be doing a lab report for this, and here's where I'm going to differ strongly from what the lab manual says. The lab report is on a single phylogeny, but I'm going to allow you to do it as a group or individual paper. It is your choice. You can either do it with your group of 2 to 3, or you can do it each of you writing your own. If you do it as a group, you're all going to get the same grade though. So this is going to be your choice, and if you decide to do it as a group paper, before you leave class on Friday, I'm going to have you sign off at your phylogenies that you have all agreed to do it as a group paper and to receive the same grade. You will need 3 references for this, 2 of which must be primary sources, and websites do not count. Journal articles, textbooks, that sort of thing is what I expect you to use. There will be in-text citations. You will need at least 1 figure, your phylogeny. Your introduction should include information about what phylogenies and cladistics are, how you build one, and then your hypothesis, which is your phylogeny. And your hypothesis might be that the phylogeny created in figure 1 is correct. In the methods, you're going to list the animals you had and the process that you went through in determining your phylogeny. In your results, your figure definitely should be there. You should explain your figure and explain your results. In the discussion section, you're going to justify the phylogeny you made. You may present alternative phylogenies. You're going to explain why the animals are where they are in your phylogeny and whether or not the literature supports this. You also may want to talk about evolutionary pathways and missing links, and perhaps some areas where there might be confusion with your phylogeny. So we'll be doing this on Wednesday and Friday during our class periods. And the goal is still to do 3 phylogenies during those 2 class periods, but we'll see how things go on Wednesday and come up with a final answer on that Wednesday. I will be, because we're doing these on Wednesday and Friday, I will be posting recorded lectures on the rest of the bird information so that we are not really losing time, and you're encouraged then to watch those during what would have been our normal lab time. I will be posting those shortly. Thank you.